Hello everyone. Today I will demo for you our diagnostic and early detection tool. I will provide a brief overview of our invention and will demo for you our method. I will also provide some definitions about the technologies, scientific methods, and tools used. I will share with you our vision and review with you the results of the live and edited demo. I apologize in advance that these slides are not presented in slideshow mode, mainly because I am using a screen recorder, and if I were to press escape to exit the slideshow mode, I would inadvertently exit the screen recorder and stop the recording of this demo. As you know, cancer is one of the leading causes of death worldwide. Early detection is the only hope for survival. Blocking the initial oncogenic transformation of cells will save lives and reduce suffering as well as reduce treatment costs. Dr. Moniz and I have worked on this invention for a long time. We were able to patent it and we have tested it on so many data sets. We are intimately familiar with all aspects of the method, and we have spent years perfecting it. Our idea stems from this profound observation. Since cancer is so heterogeneous and continues to evolve, we should use an evolutionary method, phylogenetics in this case, to process cancer data and to better understand it. Essentially, we took a well-established scientific field and applied it to cancer data. Our results were striking, and we think this invention is a major stride towards early detection. We take mass spec data or metabolomics data, which we process in our proprietary algorithm, and we feed the output of that to the phylogenetics program. The result is a tree called cladogram that depicts the placement of patients in that tree. Phylogenetics is the study of the evolutionary history and relationships among individuals, groups of organisms, or other biological entities, in our case, cancer data. A phylogenetic tree is a diagram that represents evolutionary relationships among those organisms or individuals. Here is a sample phylogenetic tree representing some species and their relationships. This scientific method is well established in zoology and botany. Our vision of the clinical application of our invention. A patient goes to a lab where they take a blood sample, then the lab processes that blood using either mass spec or metabolomics and produces measurement of molecules found in that blood. We take those measurements and run our proprietary algorithm to generate an input file for the phylogenetics programs. These, in turn, produce a phylogenetic tree representation of this patient, and we use that to derive diagnosis and early detection. The advantages of our method is that it is not restricted to a panel of biomarkers for individual specific cancers. This method is sensitive to all types of cancers. Our method looks at all biological manifestations found in the serum and provides the ability to monitor disease progression and tailor the treatment, such as the case of treatment for primary or metastatic cancer. Here are samples of phylogenetic trees of prostate cancer data that we have processed and published on. On the right side, you see that our method not only separated the healthy from cancer, but also it was sensitive enough to separate the primary from the metastatic. When we run multiple types of cancer data, such as breast, lung, colon, ovarian, etc., that have been produced using the same techniques, such as mass spec or metabolomics, we also observed that none of these cancers clustered together. These types of complete separations demonstrated the validity and efficacy of our method. On the left side, you can see this blue area without the transitional zone. In here, there are patients that are healthy, but we think they have accumulated enough mutations that bring them closer to being cancers. This is the early detection our method provides before any physical signs of cancer. To reach the scale required for a production release, we need to build a master cladogram for each type of cancer that has anywhere between 500 to 1,000 healthy patients and a similar number of patients with cancer. This is to establish a baseline that encompasses enough variation that are found within the general population. For example, for prostate cancer, we will have 2,000 men of various demographics. We will process their metabolomics data using our algorithm. And when we receive a new unknown specimen, we will add it to this universe and reprocess again. There are three potential outcomes depending where the new unknown specimen appear in the cladogram. Either healthy, if it appears here, or transitional, if it appears here, or cancers. This enables us to diagnose and achieve early detection. Now imagine we process the measurement of that same serum of a patient with the population from each cladogram representing each type of cancer. The end result is a non-invasive, holistic cancer screening with one lab visit, one drop of serum. This is the compound beneficiary factor that we can offer off the bat to our customers. I am going to demo how we analyze this data and review the result. If you would like us to analyze your data, we can do that in real time. Please contact us to schedule a meeting during which you share with us your own data without providing any labeling to us, such as healthy or cancerous. And after we process it in real time and share the result, you can be the judge. If you don't have your own data, we suggest using publicly available data sets that others have published and that are well documented that you can use for this purpose. We are seeking investors to take our invention to the FDA and after that to the marketplace. So please, if you are interested, get in touch. Together, we can produce a paradigm shift in cancer diagnosis. We look forward to hearing from you. And now, on to the demo and the review of the result. Here, I have three folders: in-group, out-group, and phylogenetics. Phylogenetics has a few phylogenetics programs that I will run and demo for you. One of them is Mix, and the other one is IQ-Tree. And this one is a tree viewer, as well as the NCBI tree viewers that we will use. In here, after I process this data, coming from in-group, which has about 79 patients, a mix of healthy and cancerous, and an out-group, 24 specimen or a species in this case, and the result of that processing is an in file. After I run the program, it will appear here in this folder. So let's get started. Here's the in file. We can click mix. Yes. While mix is running, I will take a copy of this file. And I go to this other program where I paste it. Let's rename it txt in this case. And let's run the other program. There are two phylogenetics programs. Each one uses different method. In this case, IQ3 uses maximum likelihood, and MEX uses parsimony. And before this program runs, it evaluates multiple models and picks the best model for this data set. Some definitions for you, because as you know, in these data set here, we are using metabolomics data, and you can see we have a large number of readings in for each patient. It's 4,497, and metabolomics is an emerging field and is broadly defined as the comprehensive measurement of all metabolite and low molecular weight molecules in a biological specimen, while mass spec or mass spectrometry is an analytical tool useful for measuring the mass to charge ratio m/z of one or more molecules present in a sample. The difference between these two methods, the MEX and parsimony versus IQ3 maximum likelihood, is that the method of maximum likelihood seeks to find the tree topology that confers the highest probability on the observed characteristics of the species, while the method of maximum parsimony seeks to find the tree topology that requires the fewest changes in character state to produce the characteristics of those species. Please know that at the core of our invention or our invention is our unique background. Dr. Moniz has a PhD in biology and did his dissertation in phylogenetics and worked for over 15 years in the lab of pathology at the National Institute of Cancer, NCI. I studied 2 years of biology before switching to computer science and getting a bachelor and a master's degree in the field. I have a strong background in data analytics, predictive modeling, machine learning, and artificial intelligence. And I also worked at the lab of pathology for over 3 years, where I met Dr. Moniz and started working on this with him in our free time. Here I see that the mix has finished running. Let's see the output. Okay. The out file where we can see the trees. Okay, the mix produced 4 trees. We will shortly examine. Usually they are almost identical with minuscule variation of the placement of the patient. And to do that, let's have a visual. Okay, and we can file, open the out file, tree file in this case. Doesn't matter what we call it. So this is the first tree. If I step from one tree to the other, you can see as this panel changes the variation within those trees. Keep in mind, this is all cancerous. They are donated as PBR from here, this side, this right here. All PBR, and these are healthy. And also keep in mind, observe that the healthy is the closest to the base. You see, closest to the base, and the farthest away from the base is this clade right here with all cancerous together. PBR is patients before radiation, while here is healthy. So as I step through, the variation is minuscule, just placement within the major clade, no switching sides. The same thing for tree number 3 and the same thing for tree number 4. Since NCBI viewer only accept this Newick form, I will basically extract this one, and they just call it Newick, so we will not mix it. And we can close out of here and go to NCBI. We can close out of this program. And in here we can do upload. By the way, NCBI does not display 4 trees. It displays only 1 tree at the time, and as a result it will give us an error. To avoid all of that, let's just go ahead and basically keep only one tree. I think the difference between the two is the semicolon, or between the trees is the semicolon, the separator. Okay, so we can keep the first tree. And we can use that to view here. Okay, here we go. We can change the format or the layout. Standard diagram. This is PBR. Okay, and you can see the farthest away from the base is patients before radiations, all cancer, no mix and match. And here, the closest to the base is healthy. Okay. By the way, you can basically rotate the upper hand to join if you want to, and they will be identical tree in terms of phylogenetics. The other way to see this is the other format. This is what we saw earlier. So this is the PBR farthest away and by themselves, and this is the healthy. Now let's see the other program, whether it's, yes, it's finished as well. So we can close this one, and go to the last program. Sorry, this one. In this case, this file gives you the summary of what the program did, the evaluation of the several methods, the application of the method of choice. In this case, it outputted one single tree. And let's go ahead and examine it on this side in CBI. So go back, upload. Tree file, upload, close. So it's a different tree, but with a different program or from a different program, but with no mix and match. Complete separation. Again, the farthest away from the base is the cancerous. The closest to the base is healthy, and healthy clustering together and cancerous clustering together. Complete separation. I hope this demo intrigues you, and if you are interested, please get in touch. Thank you.